Hello everyone, I am Dr. Suresh, Faculty of Biochemistry, and in today's session we will be doing a recall session about the Inicet examination that has been conducted a few days back. And first of all, before starting the session, I would like to convey my thanks to the students who have recollected the questions from the exam and posted on various social platforms like Facebook and Insta, because they are the source of this recall session. So that's why I would like to convey my thanks to them. And again, I would like to congratulate all the students who have successfully attempted the Inicet exam. So don't bother about the result. You have given your hard work. Whatever the hard work you have did, that is going to be paid off. Just have little patience and let us progress with our recall session. So to begin with, the morning session, so the first question is image based question. So here image based question, what they have asked, identify the following image. So what is this image? Image is a tonsillitis. So what is a tonsillitis? So orange tonsillitis, you can make out with the picture, right? So the options has been given as Ullmann's disease, Gaucher's disease, Tanguay's disease and Tay-Sachs disease, right? These are the probable answers students who have recollected and posted. So with that I have made it these options, right? So what you have made, like here the deposition of cholesterol on extrahepatic tissues, right, like in tonsils. So what to say? So orange tang. So what you can observe here, orange tang. So it is a clinical feature of Tanguay's disease. It is a clinical feature of Tanguay's disease, right? So and what is the thing? What is the defect? What is the defect causing this orange tonsillitis? So here the defect is the mutation in ABCA1 gene mutation. So what is this ABCA1? It is ATP binding cassette transporter protein, okay, which is helpful in effluxing cholesterol ester and phospholipids from inside the like outside the cells to inside the cells. So what happened if this transporter protein absent because of the mutation in this ABCA1 gene, the cholesterol esters and phospholipids will not be effluxed into the cell and they will be deposited outside the cells, right? So that is the reason you are getting this here. And other features of Tanguay's disease, there is decrease in HDL concentration and also hepatosplenomegaly, right? And what to say, mononeuritis multiplex, okay. Mononeuritis multiplex is also one of the prominent feature of Tanguay's disease, right. So what to remember? Mutation in ABCA1 gene which causes this Tanguay's disease. So orange tang or orange tonsillitis, you can make it. And what about other options? So Ullmann's disease. Ullmann's disease is because of deficiency of the enzyme acid phosphatase, acid lipase, right. So what is a clinical feature if they want to make a question of Ullmann's disease, like adrenal calcification, right? They will give you the picture of adrenal calcification. They will ask what is the enzyme, what is the disease. So adrenal calcification is a prominent feature of Ullmann's disease. And Gaucher's disease is because of beta glucosidase enzyme, right? Gaucher's disease, beta glucosidase enzyme. So it is like what to say, tissue paper cells. The prominent feature here is tissue paper cells, right. And Tay-Sachs, again it is like lipoprotein storage disorder. Tay-Sachs, Gaucher's, all are lipoprotein storage disorders only, right. So in that condition there will be decrease in LDL concentration. So moving to the next question, glycogen phosphorylase activated directly or indirectly, okay. The question has been asked, glycogen phosphorylase activated directly or indirectly by what substance? So options, probable options according to students, calcium, glucose 6-phosphate, insulin and glucose, right. So here according to me, like what you say, what is glycogen phosphorylase? Glycogen phosphorylase is the enzyme of, it is the enzyme of glycogenolysis, okay, to break down the glycogen, right. If you are breaking the glycogen, what are the products? You will be getting glucose 6-phosphate and glucose, right. So here we have already learned in enzymes that feedback inhibition or end product inhibition. So here the end products are glucose 6-phosphate and glucose. So here you can rule out two options. Glucose 6-phosphate is the end product of glycogenolysis, so it will not be acting as an activator, and glucose is also you can rule out, and leftover options is insulin and calcium, right. Insulin is again, it is a hypoglycemic hormone, right. So because here glycogen phosphorylase is activated to increase your glucose concentration, right. But what is the role of insulin? Insulin is a hypoglycemic, so it is also nothing to do with this option. So leftover thing is calcium. Why? So especially muscle glycogen phosphorylase activated by calcium, okay. Otherwise, like you can say cyclic AMP, this is another option. If they have asked, cyclic AMP is also be the better answer compared to calcium, okay, especially muscle glycogen phosphorylase. So why? Because in muscle calcium will be releasing from sarcoplasmic reticulum to in like excitation contraction of muscles. So that time muscles they require energy. So what happened, to get the energy, the released calcium will stimulate glycogen phosphorylase to break down the glycogen to release glucose 6-phosphate and glucose, which will be entering into glycolysis to provide ATP. So that is the purpose. So that's why answer is calcium. So coming to the third question, burnt sugar smell of urine is because of the defect of. So here again options are there: phenylalanine hydroxylase, fumarate acetoacetate hydrolase, isovalryl CoA dehydrogenase, branched chain keto acid dehydrogenase. So 4 options has been given. So burnt sugar. So we have already learnt in amino acid metabolisms, like inborn errors of amino acid metabolism, maple syrup urine disease, which has got burnt sugar smell, right. And the key enzyme of branched chain amino acid, that is branched chain keto acid dehydrogenase. This is the correct answer. And why this is the correct answer? Because the defect of this enzyme leads to accumulation of branched chain amino acids such as leucine especially. So leucine is ketogenic, right, which produces ketone bodies, and these ketone bodies start excreting in the urine, which gives a smell, burnt sugar smell of the urine of the okay. And rest of the things like phenylalanine hydroxylase, I am just giving you examples further. So it is mousy odor, right. And fumarate acetoacetate hydrolase, this will be giving you cabbage odor, right. Type 1 tyrosinemia, okay. The enzyme fumarate acetoacetate hydrolase will be in case of the deficiency of this enzyme seen in type 1 tyrosinemia, and this give you cabbage odor. And third one, isovalryl CoA dehydrogenase, it is you can what the odor here you can get? Sweaty feet odor, sweaty feet odor. So different, here it is burnt sugar odor, you see here burnt sugar odor. So if they would have asked the questions like shuffling, right. So phenylalanine hydroxylase enzyme deficiency gives you mousy odor, fumarate acetoacetate hydrolase type 1 tyrosinemia gives you cabbage odor, okay. Isovalryl CoA dehydrogenase will give sweaty feet odor, and branched chain keto acid dehydrogenase give you burnt sugar odor. So the correct answer is D, okay. And fourth one, cystic fibrosis is caused by which of the following deletion? Cystic fibrosis, there are 700 mutations have been identified as a reason for cystic fibrosis. Remember, there are 700 mutations have been identified as a reasons for cystic fibrosis, Okay, then which might be the one of the reason for out of the options like tyrosine deletion? This is example for deletion mutation. Okay, how come this deletion mutation? Because the condition is autosomal recessive. Okay, the condition is autosomal recessive, and why this is happening? Okay, there is a deletion, there is a defect of protein. Okay, that is cystic fibrosis. What is that? CFTR protein. Okay, because of defect of protein, CFTR protein. What is full form? Cystic fibrosis transmembrane regulatory protein. Okay, cystic fibrosis transmembrane regulatory protein, and because of mutation, okay, this CFTR defect leads to cystic fibrosis. Because of this, what happened? There will be inability in transportation of chlorodyne. So what for chlorodyne requires? Chlorodynes required for secretion of like what to say, sweat and gastric juices for making. So for all these purposes we require chlorodynes, right? If this cystic fibrosis transmembrane regulatory protein is not available, there is a defect in transportation of chlorodynes. So what happened in that condition? Your chloride concentration in sweat will be high. Okay, your chloride concentration in sweat will be high. So the answer here I can say that is phenylalanine at 508. Okay, so rest of the answers are wrong. Okay, tyrosine at 580 is not the correct one. Phenylalanine at 508 is the correct answer. Tryptophan at 508 is also wrong. Lysine is 805 is also wrong. So that is the significance. Okay, cystic fibrosis. Okay, because of the defect of the gene which produces CFTR protein, that is cystic fibrosis transmembrane regulatory protein, and it is because of deletion mutation at 508. Okay, deletion mutation. What amino acid has been deleted here? Phenylalanine at 508 position. So coming to the fifth question, significance of HMP shunt except. So what for HMP shunt we have learned in carbohydrate metabolism, HMP shunt significance. HMP it is required for NADPH, yes. Okay, and it is a source of ribose 5-phosphate, yes. It is a source of pentoses, and it is a source of acetyl-CoA. How? Because we have seen that 3 molecules of glucose involved in HMP shunt. So out of this, only 2.5 glucose units will be utilized, and half glucose molecule will be converting into acetyl-CoA. I mean, how it will be half molecule? Half glucose is a 6 carbon compound, right? So 3 into 6, total 18 carbons are involved in HMP shunt. But in HMP shunt, only 15 carbons are utilized. And what about the fate of 3 carbons? So the leftover 3 carbons, that will be converted to glyceraldehyde 3-phosphate, and this glyceraldehyde 3-phosphate enter into glycolysis to form pyruvate, and this pyruvate can be forming acetyl-CoA. Yes, it may be served as a source of acetyl-CoA, right? And utilizes ribose. How come? It is utilizing glucose to produce NADPH, to produce ribose 5-phosphate, it is to produce acetyl-CoA. So this option is correct, because here you have asked the question except. So rest of the things are correct with the HMP shunt significance. And sixth question, sorry, here there is a spelling mistake. Refsum's disease caused due to defect of. What to say? Refsum's disease caused due to defect of. Refsum's disease, oh, if you recollect, what for Refsum disease? Refsum's disease because of defect of branched chain, because it is a disease related to branched chain fatty acid, that too alpha oxidation. See, beta oxidation of branched fatty acids we have not seen, okay. Omega oxidation of branched chain and fatty acids, okay. Alpha oxidation of dicyclic acid, this is a ruled out option, right. So the correct answer is second one, Refsum's disease caused due to defect of Refsum disease caused due to defect of alpha oxidation of branched chain fatty acids. Okay, alpha oxidation of branched chain fatty acids. Seventh question, SARS-CoV-2 diagnosis is done by all except. So what is this SARS-CoV-2? So because of this pandemic, everyone come across with this, right? What is the test for SARS-CoV-2? RT-PCR. What do you mean by RT-PCR? PCR, n number of types are there. Real-time PCR is there, right? Reverse transcriptase PCR is there. So options here, what he has asked, SARS-CoV-2 diagnosis is done by all except what? Here RT-PCR is also correct, okay, real-time PCR, and R.T, that stands for reverse transcriptase PCR, that is also correct, and Southern blotting. So when you have come across the techniques in molecular biology, Southern blotting used to detect DNA, okay, and immunoassay. Immunoassays is for DNAs and RNAs, right? Actually to say, it's like for proteins, you can say anything, DNA, RNA, protein, everything can be done by immunoassay, right? But here SARS-CoV-2 is what? It is a virus. So what is the genetic material in the virus? RNA, right? Not DNA, right? So with this small, little logic, we can rule out the option, or you can eliminate what is that exception. So that is Southern blotting. Southern blotting is for DNA, right? So that is the reason we are using the technique reverse transcriptase method to identify the virus, right? RNA. So Southern blotting is for DNA, so it cannot be used for diagnosis of SARS-CoV-2. And eighth question, selenocysteine is present in which of the following enzyme? Glutathione reductase, glutathione peroxidase, thioredoxin peroxidase, thioredoxin oxidase, right? So selenocysteine, if you recollect, it is one of the 21st amino acid. Selenocysteine and pyrrolysine are the new inventions. Selenocysteine, there are codons which has been discovered for coding of this amino acid, right? Selenocysteine. So and selenocysteine, remember the key feature, selenocysteine, it requires for three enzymes in our body. One is glutathione peroxidase, other one is thyroxine reductase, other one is deiodinase. So these are the three enzymes which totally depend on selenocysteine. So out of these options, what you can say? Glutathione reductase, there is no option. Glutathione peroxidase, yes. Sorry, excuse me. Thyroxine peroxidase, ruled out option. Thyroxine oxidase also ruled out option. So glutathione peroxidase, okay, contains selenocysteine. So option B is the correct. True about citrulline. Ninth question, they have asked citrulline. What is citrulline? Citrulline, we are all aware, it's a substance present in yeast, right? It's a silent mating type information regulatory protein, okay? It is a silent mate information regulatory protein. It is present in red wine also, okay? So here the main thing about citrulline is, though it is a single protein, it has got two activities, okay? Citrulline, though it is a single, it has got two enzyme activities. Right, one is ADP ribosyl transferase function. What is that? ADP ribosyl transferase function, and other one is histone deacetylase function, So this histone deacetylase, what it will do? By doing deacetylation, it condenses the chromatin, right? So this mechanism helps in DNA repair. Right? And ADP ribosyl transferase, again for cell synthesis, new cells formation, right, for that purpose, and it causes what? Delay in aging. right. There are 7 types are there, okay. There are 7 types of citrulline you can found, T1 to T6, and what they do, they even increases glucose tolerance. So what are the options it has been given? Increases insulin sensitivity, yes, it increases insulin sensitivity, correct, because as it is increasing glucose tolerance means increasing insulin sensitivity. It is a histone deacetylase and chromosome condenser, yes, it is true about it. It increases lifespan, yes, by enhancing DNA repair and by enhancing DNA synthesis, it increasing lifespan. Fourth one, stimulated by niacinamide, no. So this is option you have to rule out, right? This is option you have to rule out. So rest of the 3 are correct about citrulline, but except the fourth option, stimulated by niacinamide, because niacinamide is acting as an inhibitor of citrulline. Yeah. Another thing to the morning session question, right sequence of converting cholesterol to testosterone. Okay, we are all aware cholesterol is a 27 carbon compound, right, and testosterone is a 19 carbon compound, right. So how to condense 27 carbon cholesterol to 19 carbon? So what are all the enzymes will be involving in that, right? So when you talking about cholesterol, cholesterol is 27 carbon, right, and it will be converted to pregnenolone. I will write in this direction, okay. Okay, cholesterol is a 27 carbon, it will be converting to pregnenolone, which is a 21 carbon, right, and this pregnenolone again converted to progesterone, which is again a 21 carbon compound, and again this will be converted into 17 hydroxy progesterone, which is a 19 carbon compound. What to say? 17 hydroxy progesterone, it is a 21 carbon again, right, and this 17 hydroxy progesterone converted to androstenedione, which is a 19 carbon, right, and this androstenedione converted to testosterone, which is a 19 carbon compound, and this testosterone further removing one carbon, it will be converted to estrogen, estradiol and estrogenesis. Okay, these are 3 compounds related to estrogen, right. So now we will see what are the enzymes here. So converting cholesterol to pregnenolone, so the enzyme involved is cholesterol desmolase, that is 20-22 desmolase, okay, 20-22 desmolase, and pregnenolone to progesterone 21-3 beta hydroxy steroid dehydrogenase, and now converting progesterone to 17 hydroxy progesterone, there we require hydroxylase, right. Now converting 17 hydroxy progesterone to androstenedione, there we require 17-20 lyase, right. You see with the itself you can make out, right. So the sequence, the first one 20-20 desmolase, 3 beta hydroxy dehydrogenase, 17 alpha hydroxylase and 17-20 lyase. So this is the correct sequence, the rest of the sequences are wrong, right. So converting 27 carbon to 19 carbon testosterone, right. So this way you have to remember, first it will be coming to 27-21, 21-19, okay, that is androstenedione, and androstenedione will be converting back to testosterone, okay. and this testosterone again further removing one more carbon becomes estrogen 18 carbon compound. The 11th question, 2,3-BPG causes unloading of OT to tissues because what's happening? So why 2,3-BPG causing unloading of oxygen to tissues? The reasons has been given: decreased ATP production, increased ATP production, binds to central cavity of hemoglobin surrounded by histidine and lysine favoring salt bridges. Fourth one is inhibit TOT structure. Here 2,3-BPG, so if you see the structure of hemoglobin, so this is the structure, if you see, make out, this is the diamond shaped structure, right, at the center when 4 subunits of hemoglobin join. Okay, this is alpha, alpha 1, beta, beta prime, right. So 2 alpha, 2 beta units. So here at this point 2,3-BPG will go and bind. Okay, 2,3-BPG, 2,3-bisphosphoglycerate will go and bind, and it favors the TOT conformation of hemoglobin. Okay, there are two things, okay, TOT and relaxed conformations of hemoglobin. TOT favors deoxygenation, whereas relaxation form confer oxygenation, right. So here what it is doing, 2,3-BPG, okay, it is going to form TOT, okay, it is not going to inhibit TOT structure. So one option we have ruled out, right, and it is nothing to do with ATP production. Okay, so first two options also eliminated. Leftover is binds to central cavity of hemoglobin surrounded by histidine. Yes, right, here the beta chains of adult hemoglobin contains huge amounts of histidine and lysine subunits which are positively charged, whereas 2,3-BPG is negatively charged. So there is always attraction between positive and negative. So when there is 2,3-BPG, okay, as histidine and lysine are positive charge, they will attract towards, I mean, like 2,3-BPG, and they will prefer 2,3-BPG rather than oxygen. So that's why oxygen will go out and 2,3-BPG will come in to form salt bridges. So third option, binds, that means 2,3-BPG binds to central cavity of hemoglobin surrounded by histidine and lysine, which is favoring salt bridges. Okay, so that is the reason when there is oxygen and 2,3-BPG side by side, hemoglobin will prefer, like suppose if hemoglobin is carrying oxygen from lungs to tissues, a tissue because of glycolysis there is a Rappaport-Liebring cycle, because of that 2,3-BPG is there. So at the moment hemoglobin, I mean oxygenated hemoglobin reaching near to the 2,3-BPG, it thinks that okay, that is the one I want, not the oxygen. So it will dump the oxygen there in the tissues and will take up the 2,3-BPG, right. This is the logic behind of this, right. So the last question of the morning session, identify the left curve given the fact that right curve is of adult hemoglobin. So here this is the right curve, this is the left curve. So oxygen dissociation curve, sigmoidal. Okay, sigmoidal in shape. So when the sigmoidal curve shifts to right side what, left side what, right. Here the options they have been given, myoglobin, okay, the options they have given like myoglobin, fetal hemoglobin, copper and acidosis. So copper I can rule out easily. So it has only to do with fetal hemoglobin, myoglobin. Fetal hemoglobin further it shifts to right, not to the left, right. So it will be the myoglobin. Why? Why? Because fetal hemoglobin, it is what is the adult hemoglobin contain? Beta chains, but fetal hemoglobin contains 2 alpha 2 gamma chains, right. So these gamma chains lacks positively charged amino acids, remember, okay. So these gamma chains lacks, the difference main, beta chains are rich of histidine, lysine amino acids, gamma chains are lack histidine and lysine. So they don't have much positive charge, right? So they have to keep the oxygen with them, right? So their 2,3-BPG doesn't have any role here, right? So myoglobin, what to do? So the affinity for oxygen increases in case of fetal hemoglobin. Okay. So here the question asked is curve shifted to left. So fetal hemoglobin curve will be shifting to right side, not to the left. So it is also ruled out option. Acidosis also will not shift the curve to left side. So it is also ruled out. Leftover option is myoglobin. Why? Because myoglobin always it's like what to say, is favoring the oxygenation. So it will be shifting the curve towards the left side, and again this left side shifting of the curve, in what are the possibilities you can see in carbon monoxide poisoning, alkalosis, right? In this condition only the curve will be shifting to the left side, right? So correct answer will be myoglobin. If they have asked the option, if they in place of myoglobin, if they have asked carbon monoxide, that is also correct, right? So that's all about the morning session, recall session, morning Inicet exam recall session. Thanks for watching.